Hello, my name is Lixin Yang, and I am from the North Carolina School of Science and Math. My research focuses on the effect of small molecules JHRE06 and T2AA on mutagenic translesion synthesis in Arabidopsis thaliana. To give a little bit of background information, cancer is a genetic disease characterized by abnormal cell growth, and it is one of the leading causes of death in the United States. One of the greatest things that cancer affects in the human body are DNA repair genes. In all of our cells, we have healthy DNA repair systems that help mitigate against the damage done to DNA in everyday situations, such as UV rays from the sun or chemical radiation. But however, if these cells become cancerous, they lose a lot of these DNA repair systems, so they have to resort to an alternate repair strategy called translesion synthesis, for TLS for short. Now, translesion synthesis is very inaccurate. Instead of fixing DNA lesions that is found in our DNA, it actually focuses on cellular division instead. So if cancer cells undergo TLS, tumors can develop new mutations. They can follow a relapse after chemotherapy treatments. So the goal, if you want to improve chemotherapy success, is to find drugs to kind of target this TLS process to see if we can bring success rates back up of recovery. So if you look at a diagram of TLS inside Arabidopsis, you will notice that there are two pathways, the Rev1 dependent pathway and the Rev1 independent pathway. On the Rev1 dependent pathway, you have two family polymerases, the TLSY family polymerase and the TLS pol polymerase. And once these two both bind to that Rev1 protein, the Rev1 dependent pathway is able to continue with TLS. On the Rev1 independent pathway, you have the same two Y family polymerases, and these bond to the pCNA of this entire process. And once everything is bonded and connected, the pCNA then moves on to bind to that pol delta protein. And once that happens, TLS continues. So I looked for two small molecules that can hopefully inhibit one of these interactions in order for TLS to be inhibited through all of the pathways. And I found two, JHRE06 and T2AA. If we go back to the diagram, JHRE06 is able to inhibit this interaction between Rev7 and Rev1 by binding to Rev7. And once that interaction is cut, the Rev1 dependent pathway can no longer continue with TLS. For the other pathway, I found T2AA, the small molecule, which binds to the pol delta. Once it binds to the pol delta, it can no longer bind to the pCNA, which means that the independent pathway can no longer continue with TLS. So essentially, TLS is stopped on both pathways. So my goal for this research project was to basically investigate whether or not there was a synergistic effect in combining both drugs instead of just using one of them or the other individually. In order to do this, I had two steps to my methods. My first step was to expose my Arabidopsis seeds to UV so that they can use TLS as their main form of cellular repair. And I did this using a UV protocol where I exposed all my seeds for 15 minutes every other day for 5 total days. And once this is completed, I'll know that they're using TLS for cellular division, and the seeds will continue to germinate and grow so I can use my inhibition drugs to see whether or not I can inhibit this TLS. So for my experimental design, I have 4 treatments. The first is my negative control. There are no treatments. My second is my positive control with my JHRE06 treatment. My third has a T2AA treatment, and my fourth combines both drugs together. For each of these treatments, I have 10 seeds on each plate, and I'm using the wild-type Columbia strain for all of them. After my designs, I will practice a root bending assay and measure my plant growth to see if TLS has been successfully inhibited. My root bending assay is basically a process where I can see how long it takes for the plants to reorient themselves after being turned. So what I did was I took all the plates and I flipped it so that it was standing straight up so that the sprouts can grow vertically. After a couple of days, I will rotate the plates so that the sprouts are horizontal. Then I will see how long it takes for those roots to start reorienting themselves downwards due to gravity. And I used imaging software to measure the degree of root bending after a couple of days, and as well as root length. So these are my results. As you can see, in root length, The control had a significant difference when compared to JHRE06, T2AA individual treatments, and as well as all of them combined. Similarly, the JHRE06 and T2A individual treatments had a significant difference when being compared to the combination treatment. This is the same for root angle, where I saw a significant difference between the control and all three other treatments, as well as a significant difference between each individual treatment to the final combined treatment. However, if you look at both root length and root angle, you would see that the JHRE06 treatments and the T2A individual treatments had no significant difference when being compared to each other. You can see here the results that I have a visual representation of what each Arabidopsis root looks like. You can see in the control that since TLS is functioning and cellular division is happening, you have a lot of root bending in here. If you look at the combination treatment of both drugs combined, you can see how there's very little root bending, which signifies that it's taking a longer time for them to reorient themselves since TLS is inhibited. So in my conclusion, I see that individual JHRE06 and T2A treatments have a significant difference in TLS inhibition than compared to the control, since both drugs have similar rates of TLS inhibition. However, when we combine both treatments, the rates of TLS inhibition are significantly greater than individual treatments and the control, which signifies that combining both drugs possibly has a synergistic effect of reducing rates of TLS, which proves to be a possible path in future research about cancer and translational synthesis. Any future research done for this project will look into a computational analysis of both drugs to see really like the deep protein interactions to see what they truly do. I will also repeat this experiment with different trials, with different concentrations and model organisms to see if I can get the same response and same results for all of my trials. For my acknowledgements, I would like to thank my mentor, Dr. Monahan, and my research and biology class for guiding and supporting me through this research process. Further thanks to Dr. Sheck, the Glaxo Endowment, and the North Carolina School of Science and Math for giving me the opportunity to conduct my own research. And here are my references. Thank you.